Hello, I'm Dr. Nadia Boutaoui. I have a PhD in genetics and an MBA in healthcare. I'm here along with Dr. Ahmed Chenna, who has a PhD in chemistry and is a principal scientist in Monogram Biosciences. We put together this COVID-19 vaccine talk. The agenda includes going over the background, history of pandemics, vaccines, and coronaviruses, the vaccine development, its methods and formulation, the approved vaccines, Pfizer and Moderna, and what it would take to vaccinate many people, the challenges ahead, and prices. First, we'll review the history of pandemics, vaccines, and the coronaviruses. There were many pandemics in the 19th and 20th century, including the Spanish flu that killed 50 million people, the Asian flu that killed 1.5 million, as well as the Hong Kong flu in 1968. In 2009, there was the pandemic flu, H1N1, and 300,000 people died. And we are in the midst of an unprecedented pandemic, COVID-19, that killed 1.7 million to date as of December 21st, 2020. Vaccine development is not new. It started as early as 1921 with development of tuberculosis vaccine, all the way up to development of the Ebola vaccine in 2019. As of the past 2 weeks, we already have 2 approved COVID-19 vaccines. Coronavirus infections are not new. In the 1960s, there were reports of these viruses, SARS in 2002, MERS in 2013. They were present in many countries, with the worst being COVID-19 in 166 countries. Their lethality is also different, with COVID-19 killing 1.7 million people to date. This diagram shows the COVID-19 structure. It includes a spike protein, that's where it gets the name the coronavirus, an RNA, as well as an envelope. Next, we'll review the vaccine development, the methods and formulations. Drug or vaccine development take 10 to 15 years in a traditional timeline, where there is an exploratory phase followed by preclinical phase, then clinical trials that include phase 1, phase 2, and phase 3, followed by FDA approval prior to manufacturing. In the accelerated timeline, the clinical trials, the FDA approval, and the manufacturing, the rapid manufacturing, are stacked in a way to shorten that timeline. And that's what we've seen in vaccine development of the COVID-19. Important characteristics for the vaccine include its safety, efficacy, does it work in different target population, and how long would it protect the individual? Is it months or years? There are several types of corona vaccine methods. These include DNA and RNA, where the vaccine uses a piece of DNA or RNA molecule to teach the immune system to target the viral protein. In the live attenuated vaccine, there is a weakened version of the actual virus. In the inactivated type, the virus is inactivated by heat or other chemicals prior to injection. There's also the subunit, where there is a piece of the virus surface to focus the immune system on a single target. There's also the viral vectors. In this approach, you take a harmless virus to deliver the viral gene to build immunity. The only method that hasn't been used before is the DNA and the RNA virus vaccine development method, and those are the 2 that are done by Moderna and Pfizer. The remaining 4 methods have been used before for a successful vaccine development. For DNA and RNA viruses, they are easy and quick to design. However, they are new, so we don't know about their effectiveness in the long term. The live attenuated viruses, they stimulate a robust immune response, but may not work with an individual with a compromised immune system. The inactivated virus is safe, however, may not be as effective as the live virus. The subunit method focuses the immune system response on the most important part of the virus, but it may not simulate a strong response as a result. The viral vector, the live viruses, they tend to elicit a strong immune response. However, it's very important to pick the viral vector that is truly safe. We've seen an unprecedented race to create vaccines for COVID-19. As of December 18, 2020, there is a total of 81 vaccines in clinical trials on humans. They include 43 in phase 1, 20 in phase 2, and 18 in phase 3. There's also some that were limited and 2 as of 2 weeks ago that have been approved for emergency use, and these were both mRNA vaccines. In the next section, we'll go over the approved mRNA vaccines by Pfizer and Moderna. How do the mRNA vaccines work? Scientists focus on the spike protein. They learn how these cells produce that protein via what's called messenger RNA. They packaged that messenger RNA in a lipid that delivers these instructions to the cell, which then can make that spike protein and trigger the immune response. So what's in these vaccines and why is it there? These vaccines include the active ingredients, which is the mRNA coding or the instructions for the cell. These will trigger those cells to produce the viral protein and start the immune response. There is also lipids that can deliver the RNA to the cells and protect it from degradation. There's also some salts. These are buffers for the vaccines to stabilize its pH so it matches the pH of our bodies, as well as sugars. These sugars, the sucrose, help cryoprotect these vaccines in ultra low temperatures. The Pfizer clinical trial, phase 2 and 3, included over 43,000 people. They were 50-50 male-female distribution. They were mainly white, and they ranged from age 16 all the way to over 75-year-old. In the clinical trial, the vaccine or the placebo is given to a group of people, and after 2 doses, the COVID-19 is tested. So what they found in the Pfizer BioNTech vaccine, the vaccine group had only 9 people that tested positive for COVID, and the placebo group had 171 people. The vaccine efficacy was about 95%, and this was calculated as follows. You take the number of people in the vaccine condition, divide it by the placebo, and subtract that amount from 1, and that's how you get the efficacy of the vaccine. In this case, 0.9473. Both the Pfizer and the Moderna are mRNA-based vaccines. However, they have few differences. For doses, they both require 2 doses. For Pfizer, they are 21 days apart. For Moderna, 28 days apart. For Pfizer, it's for 16 years old and up, and for Moderna, 18 years old and up. For the efficacy of the vaccine, they're both similar, around 95%. However, for Pfizer, it's 94% in over 65-year-olds, and there is no difference in vaccine efficacy by age in Moderna. One big difference between the two vaccines is the temperature storage. For Pfizer, it requires ultra-low storage temperature, -80, and for Moderna, -25, and this can play a major role in the adoption and the distribution of the vaccines. Some of the commonly reported side effects for these two vaccines include injection site reaction, fatigue, headache, muscle pain, chills, joint pain, and fever. Notice that for most of these side effect reactions, they are higher in Moderna compared to the Pfizer vaccine, and this could be linked to the formulation to withstand differences in temperature, storage temperatures. So there were some new side effects of the Pfizer vaccines in the UK that had over 140,000 people vaccinated, and they reported allergic reactions in 4 people, which is a very small number. So as a precaution, don't take the vaccine from Pfizer's for people who have previously had allergic reactions to food, medicines, or vaccines. Other vaccines in the pipeline include that from Oxford University ASTRAZENECA vaccine that reported an efficacy between 70 and 90%. It works well in older adults, including over 70-year-olds, and it showed antibodies that last at least 6 months. Next, we'll review the challenges facing vaccinations as well as pricing. Vaccines have been used to eliminate diseases. For example, these diseases have a basic reproductive number, or R0, which is how many people can be infected by one person. For example, SARS, which is transmitted by airborne droplets, similar to COVID-19, has an R0 between 2 and 5 and a herd immunity threshold between 50 and 80%. The COVID-19 basic reproductive number is between 2 and 3. We can then calculate the herd immunity threshold as follows. It's 1 minus 1 divided by R0, and that gives us a herd immunity threshold between 50 and 67%. Here is an overview about the global vaccination coverage in the world in 2019. So it ranges from 40 all the way to almost 90%. And for COVID-19, the threshold for infection elimination is around 70%. To vaccinate 7.7 billion people, we need to develop multiple vaccine platforms, coordinate from testing all the way to delivery of the vaccine, and manufacturing at scale is key. Challenges to distribute vaccines include gaining public trust, keeping vaccines cold, building a skilled workforce, getting people to health clinics, and making sure low-income countries get the vaccine. COVID-19 prices for the approved drugs Pfizer and Moderna range from $20 to $37. For AstraZeneca and Oxford University is between $3 to $4. That's a traditional vector vaccine. J&J at $10, and Novavax and Sinovac between $16 to $30 per vaccine. Thank you for your time and for listening. You can follow us on social media. My Facebook page is Dr.Nadia Boutaoui. We can also be found on LinkedIn and Twitter. Stay tuned for other videos about COVID-19 and other science-related subjects. Thank you.